Hello friends, today we are going to learn the topic enzyme. So the learning outcomes of this topic are as follows. After seeing this presentation, the learner will be able to define different types of enzymes. He will be able to classify different types of enzymes and also describe the mechanism of enzyme activity and their application in different fields. Introduction. The enzymes are the biological catalyst which helps to speed up the rate of biochemical reactions which occur in our body. These are three-dimensional globular proteins. The enzyme, which consists of special RNAs, are called as ribosomy enzymes, and the example is hammerhead ribosomy. Classification of the enzymes. The classification of enzyme was firstly developed by International Enzyme Commission. International Enzyme Commission classified the enzymes on the basis of the reaction catalyzed. There are 6 major classes, that is oxidoreductase, transferase enzyme, ligase enzyme, isomerase enzyme, hydrolase enzyme and lyase enzymes. So let's study this enzyme classification in detail. So first class is oxidoreductase enzyme. This type of enzymes helps or catalyzes the oxidation and reduction reactions. Example is lactate dehydrogenase. Second type is hydrolase enzyme. As the name indicates, they helps in the catalysis of hydrolysis reaction. They are also involved in the bond cleavage with transfer of functional group of water, and example is lysosomy. Third class is ligase enzyme. Ligase enzyme helps in the synthesis of new covalent bonds between the substrates, and example is RNA polymerase. Lyase enzyme, they help in breaking of non-hydrolytic bonds, an example is fumarase. Next is isomerase enzyme. This enzyme mainly help in the transfer of intramolecular functional group. Example is triose phosphate isomerase enzyme. And the last class is transferase enzyme. As name indicates, these transferase enzyme help in the transfer of chemical group. Example is hexokinase. Dear learners, now please pause the video and think that how enzyme helps to convert the substrate into product. Please note your answer in your notebook. After completion of the answer, please continue the video. I hope you all have noted your answer. So, the main mechanism of conversion of substrate into product mainly involve the binding of the substrate to the active site of an enzyme. This is very important step for the conversion of substrate into the product. When the substrate comes in contact with the active site of enzyme, the substrate gets bind to the active site of enzyme with the help of weak bonds, for example hydrogen bonds. This enzyme substrate complex undergoes some changes. The enzyme brings about some changes in the substrate molecule and converts it into the product, and the enzyme product complex is formed. When the bond breaks, the product is released and the free enzyme with its active site is available for the further reaction. Components of enzyme activity. As we have seen in above slide, the active site of the enzyme and substrate are the essential component for the conversion of the substrate into the product. The enzyme active site is the portion of the enzyme to which the substrate binds. This binding is due to the weak ionic bonds. Substrate is nothing but the reactant in the biochemical reaction. Applications of enzyme. So first application is that the enzymes are used as therapeutic agent. For example, the streptokinase enzyme, it helps to clear the blood clots. Second application is they are used as analytical reagents. The glucose oxidase or peroxidase enzyme is used to estimate the blood glucose level in the given sample of blood. Third is immobilized enzymes. Their enzymes are also used as diagnostic agents. They help in diagnosis of different disease. For example, elevated level of lactose dehydrogenase enzyme, it indicates the possibility of heart attack. So friends, I hope you might have understood the mechanism of enzyme activity, application of enzyme activity, and its classification of enzymes. So here we conclude our session. Thank you.